Hello friends, myself Professor Shantaya Gurmat, Department of Botany, MGVC Arts, Commerce and Science College, Mudebihar, Karnataka State. Friends, today's class we discuss about part 4. Pteridophytes, the best example is Selaginella. Example Selaginella, it comes under division Lycophyta, class Ligulopsida, order Selaginellales, family Selaginellaceae, genus Selaginella. Then we discuss about distribution. It is a widely distributed genus with 700 species found under shady and damp places, especially in the tropical regions of the world. They are rare in the temperate regions and almost absent in the alpine zones. 71 species of Selaginella grow in the tropical and subtropical forest along with the Himalayas and in the plains of India. Then we discuss about the external structure of Selaginella. Many species of Selaginella are herbaceous, perennial, few are annuals. Selaginella pygmea. Majority are dorsiventral, grow prostrate. The best example is Chrysocalis and Selaginella pallidismia and Selaginella kraussiana. A few are radial and grow erect. Then we discuss the stem. Stem is herbaceous, branched, solid and may be prostrate, sub-erect, climbing or erect. Stem develops and divides dichotomously into two equal limbs that bear spirally arranged isophilous leaves. The two limbs of the first dichotomy again branch dichotomy and ultimately monopodial. See friends, this is the external structure of Selaginella plant. Then the strobilus, here the rhizophore. The strobili develop at the tip of stronger branches, whereas the rest remains sterile. Then leaves. The leaves are microphyllous. Each leaf is traversed by a single unbranched midrib. The leaf traces do not leave any leaf gaps in the stem stele. A ligule arises from the base of each leaf, that is ligulate. Then we discuss about the ligule. A ligule develops quite early during the ontogeny of the leaf and arises from the base of the upper side. It varies in shape. Different species of the genus. The ligule may be tongue-shaped, wide-shaped, and wide-shaped in Martinsea and Selaginella and lobed in Selaginella caulescens. A mature ligule has a prominent basal portion called the glossopodium. The glossopodium is sunk in a definite ligular pit or protect its function is not well understood. Then we discuss about the rhizophore. It is a structure controversial morphological nature and present in most of the dorsiventral Selaginella. They are leafless and positively geotropic organs that have usually a localized organ and develop from the single meristem. Then we discuss about the root. Roots are adventitious and originate from the tip of rhizophore, from the solan base of the hypocotyl and directly from the stem. Arise endogenously and branch dichotomously. Then we discuss about the internal structure of Selaginella. First we discuss about the TS of stem. Transverse section of Selaginella stem shows three regions: epidermis, cortex and stele. First part is epidermis. It is made up of single layer of thin-walled rectangular or barrel-shaped cells and is covered by a thin layer of cuticle. The cells are colorless. There is no stomata. The next part is the cortex. It consists of many layers of cells. The outermost layers, 2 to 4 cells, develop thick walls in the older regions of the stem and forms sclerenchymatous hypodermis. The rest of the cortex is made up of thin-walled chlorenchymatous, means parenchyma cells contains chloroplast, and polygonal cells. Air space next to the cortex is a large air space in the center of which lies stelae suspended by means of trabeculae. The trabeculae possesses Casparian strips. See friends, this is the TS of Selaginella stem. The last part is the stele or vascular region. There is a single central stele suspended in the airspace by means of the trabeculae. The stele in this species is flattened like a ribbon and is called a protostele because there is no pith in the center. A single layer of thin-walled cells called the pericycle. The center of the stele is occupied by the xylem tissue. The xylem therefore forms a core of the stele, there being no pith. The xylem consists of metaxylem and protoxylem. Protoxylem is monostelic because the stem has only one such stele. Other species such as stele are Selaginella martensi and Selaginella chrysarides and the flabellata. In Selaginella wildernovi, there are 3 or even 4 ribbon-like protosteles. The xylem is egg-arch. There is 2 groups of protoxylem, one at either end. In the central stele varies in the outer that 2 lie on either side of it. There are more than 2 groups of protoxylem. The xylem is surrounded by phloem. The pericycle is made up of 2 to 4 layers of thin-walled cells. In this species, the stele at the base of the stem is monostelic and becomes tristelic in the middle regions of the stem. See friends, this is the TS of Selaginella stem. Next, in the TS of rhizophore. In transverse section of rhizophore, a single layered epidermis whose cells may be thick walled. The cortex is extensive and is usually distinguished into an outer thick walled sclerenchymatous cortex and the inner thin walled parenchymatous cortex. The last layer of the cortex is called the endodermis and is followed by a single layer parenchymatous cells called the pericycle. The stele is typically protostele and shows variation in its form and arranged from the protoxylem in the different species of Selaginella. Then we discuss about TS of root. Internal structure of root. Epidermis is a single layered and is covered by a thin cuticle. Root hairs are present. The cortex is wide and extensive and usually consists of an outer sclerenchymatous cortex, 3 to 4 or more layers of thickness. The endodermis is usually indistinct, but in some species it is distinguishable. Below the endodermis is a single layer pericycle. The stele is protostele. Lastly, we discuss about the internal structure of leaf. It is a dorsiventral and consists of a distinct upper and lower epidermis that are one cell in thickness. The stomata may be present on both the epidermal layers. The mesophyll is composed of thin-walled cells that are loosely arranged and encloses small or large air space. The vascular bundles is very simple and surrounded by a single layer of phloem. See friends, the first one is this is the transverse section of rhizophore. Second one is the transverse section of Selaginella root. The TS of leaf. Then we discuss about the ligule. So once again we discuss ligule. Ligule arises from several short rows of Superficial cells, a fully developed ligule consists of a distinct and hemispherical basal region where cells are large and thin walled and contain vacuolated cytoplasm. The region is the glossopodium and is surrounded by a sheath called the glossopodial sheath. Then we discuss about the reproduction in Selaginella. In Selaginella, there are two important methods. One is vegetative method and another one is by spores. The first method is by vegetative propagation. In this method, first one is fragmentation. It is effected only in species that grow under humid conditions, develop adventitious branches, and later serve their connections from the parent plant and grow into separate individual plants. The second method is tuber formation. Formation of tuber has been reported by number of species. They are formed underground favorable conditions. The tuber germinates to produce new plant in Selaginella. Lastly, we discuss about resting buds. They have been reported to develop at the ends of some aerial branches in Selaginella criscolus. Then in reproduction by spores, Selaginella is a heterosporous and the spores are of two kinds, microspores and megaspores. They are produced in microsporangia and megasporangia. First we discuss about microsporangia. A microsporangium may be oval, ovoid, reniform or spherical in shape and varies in color from red, yellow to brown. It is smaller in size than the Megasporangium. It has short multicellular stalk that varies in different species. The wall of the Microsporangium is two-layered thick. The inner wall is the tapetum, which is nutritive in function and arises from the archisporal tissue. The wall layers and the tapetum encloses a large number of microspore mother cells. The microspore mother cells are diploid and are last cells of the diplophase or sporophytic generation. These undergo meiosis to form the numerous microspore tetrads. Then we discuss about the Megasporangium. It is comparatively larger in size, four-lobed, shortly stalked, and may be green or cream colored. It has two wall and a third innermost layer called the tapetum. The functional megaspore mother cell undergoes meiosis to form tetrad of megaspore, four haploid megaspores in the Megasporangium. See friends, this is the longitudinal section of Selaginella stobilus or cone. These two are the first one is the mature Megasporangium and the second one is the mature Microsporangium. Then we discuss about the Megagametophyte. Microspore germinate to produce Megagametophyte. The antheridium of the reduced male gametophyte, the spermatozoid has three parts, nucleus, vesicle and motor apparatus. Then Megagametophytes. It develops as a result of germination of megaspores. The mature archegonium consists of a distinct neck composed of two tiers of four cells each. The upper tier functioning as cover cells. The venter portion consists of single ventral canal cell and an egg cell. Then we discuss about the fertilization. The Megagametophyte falls on a moist soil and the Microgametophyte enclosed within the spore wall happens to fall on it and discharges the spermatozoids, swim in dewdrops or rainwater, and each of the open archegonial neck, more than one sperm enters the neck and fuses with the egg to form a zygote. The last part of the Selaginella is life cycle. It is sporophytic plant and produces two different types of spores, microspores and megaspores. In other words, it may call it as heterosporous plant. These spores germination produce male and female gametophytes respectively, which in turn develop upon the stobulus and present produce the antherozoids and an egg. Antheridae and Archegonia fuses respectively. These reproduce structure after fertilization produces zygote, which again on germination gives rise to a sporophytic plant. In this way, the sporophytic and gametophytic generations alternate with each other, although the sporophytic phase is dominant and gametophytic generation is restricted. See friends, this is the life cycle of Selaginella. Selaginella plant itself is sporophytic and it produces heterosporous because it produces two kinds of spores. One is Microsporangium and Megasporangium. Both are developed in a same stobulus. Microsporangium is smaller and Megasporangium is larger. Microsporangium produces the microspores and Megasporangium produces the megaspores. Each germinate to produce the microgametophyte and megagametophyte. And there is a development of Antheridium and Archegonium. Antheridium produces the spermatozoids or sperms, and the Megagametophyte produces the Archegonium. The female gamete is egg. Both are united to form the zygote. Then the zygote by further division to produce the young Selaginella plant. This is about the life cycle of Selaginella. Okay. Thank you friends. We meet in the